Next slide. You can see here what we've got across the x-axis is the uncoiled cochlea. You can see it's a little over 30 centimeters long. And the amplitude of response, movement, and in response to what pitch. So you can see near the base, the basilar membrane moves a lot to 16,000 hertz, but it also moves quite a bit to, looks like about 10,000 hertz. That's because the membrane is a continuous piece of tissue. One part can't move without other parts moving. So let's look at how lateral inhibition would work. So you can see what I represent here is, let's say there's a hair cell right on the spot that would move in response to 4,000 hertz. That's a fairly high pitched sound. So that hair cell would be bent as it moves to 4,000. Now the hair cell on either side would also bend. But here's what happens. The hair cell that is bent the most, the one at 4,000, excites its primary synapse. So this would be a neuron that sends a signal at the auditory nerve. So the let's call it 4,000 hertz hair cell is excited, and synapses exciting that neuron, thus the big plus. Simultaneously, that neuron would inhibit the primary neuron for the hair cells on either side. So as it responds to 4,000, it simultaneously shuts down 3,900 and 4,100. This would extend out, and similar lateral inhibition would shut down 3,800, 4,200, and so forth. So what happens is you get much more responding from the peak stimulation, the exact frequency to which the hair cell responds the most, and it simultaneously inhibits responses from all the hair cells on the basilar membrane in nearby areas that are responding just because the basilar membrane moves as a single unit. So the signal that comes to A1, that is the 4,000 hertz hair cell, ends up being tuned or made more specific by lateral inhibition to the surrounding cells at this first critical synapse. This is all happening in the ear, in the basilar membrane. Next slide. This is just a different way to illustrate the same thing. You can see on the left side, lateral inhibition represented by lots of neurotransmitter released from neuron B, and this is in response to tactile stimulation, looks like pain. Not very much neurotransmitter released from neuron A, or receptor A or C. Thus, there are fewer dots between A and its first neuron, C and its first neuron. But then B goes even farther. It actively inhibits neurons at the next, neurotransmitter at the next synapse. So B sends a signal through its primary pathway, but the pathway for A and C actually have no response. That would be lateral inhibition. It's represented on the right side by showing the frequency of action potentials. And you can see that B would have the most frequent potential, most frequent response, A and C much less. And you can see with the lateral inhibition, B really pops out as different from the neurons on either side or around it. Next slide. Okay, next slide. This is a lateral view of the brain, and you can see a couple of areas we've talked about before, Broca's area and Wernicke's area. But what I want to draw your attention to now is the purple spot, and here it is called the transverse temporal gyrus of Heschel. We'll just call it Heschel's gyrus, or area 41. This is primary auditory cortex. Technically, area 41 works closely in collaboration with area 42, and we actually refer to primary auditory cortex as A1. So that's auditory 1. You can see where these are located. They're on the temporal lobe, up where the cortex and lobe roll down into the lateral fissure, the thumb of the boxing glove. So those are the structures that I want you to think about. Next slide. Here they are represented a different way. You can see that the temporal lobe here to the left, you can see T1 and T2, those are temporal lobe areas, and the red arrow indicates, it says gyrus de Heschel, that's because it's French, but it's Heschel's gyrus, area 41. So primary auditory cortex is down inside that lateral fissure. Above that is parietal lobe, below that is temporal lobe, and then you can see some of the other structures we've talked about. Corpus callosum, the thalamus, amygdala, hippocampus, and way down at T4, the fusiform facial area, which we talked about in the context of vision. So primary auditory cortex is down inside the lateral fissure, part of the temporal lobe. Next slide. So you can see here the retractor in the illustration on the left, the retractor is pulling back the frontal lobe so that you can look down inside the lateral fissure, and there is Heschel's area, primary auditory cortex. And the first thing you'll see is that it is mapped by frequency. So at one end, if that part of the cortex is responding, it means you're hearing 1600 hertz. At the other end, if that part of the cortex is responding, you're hearing 500 hertz. This would follow the law of specific nerve energies. The axons coming into the area for 16,000 hertz would be neurons from the very base of the basilar membrane, and so forth. So the way the brain is organized corresponds to the way those nerves come in from the inner ear. Okay, next slide. So that would explain to us how we perceive the pitch of sound. Now the next question we're going to consider, how do we perceive the location of a sound? So let's consider next slide. So now let's consider this illustration. The participant is staring straight ahead, and the sound source is coming from the participant's left at about 10 o'clock, 10:15, if you imagine a clock face. The two red arrows indicate the direct line from the sound source, say a speaker, to the ear. And what you see is that the two red arrows are different. That is, it is farther to the right ear than it is to the left ear. Necessarily, if the sound has to travel farther, it will arrive at one ear before the other. This slight difference in time of arrival, known as interaural time difference, ITD, is important. It tells us if it comes to my left ear before my right ear, then it must be on my left, and vice versa. It turns out there's a second important attribute or characteristic of this sound. Notice if it comes to your left ear, it passes through only air. But if the sound comes to your right ear, it has to deal with the head in order to get to the ear. It either wraps around it or tries to pass through it. In either case, that's going to have an effect on the loudness of the sound. if the sound has to pass through or around the head, it will be reduced in intensity. Thus, there's an interoral time difference. There's also an interoral loudness difference. Again, if a sound is louder in the left ear than the right ear, then it must be on the left. Next slide.